Hello everyone, myself Dr.Anvesh Vemula. I am here to explain the internal as well as external structures of eukaryotic cells. As we know, eukaryotic cells are majorly comes under the kingdom Protista, then Animalia, Plantae, and in case of eukaryotic cells, it contains beneath the cell membrane, it contains cytoplasm. So this cytoplasm is said to be smaller portion when you compare with the prokaryotic cells, and this eukaryotic cell contains the central portion of the cytoplasm is of nucleus, and many organelles are present in the cytoplasm of this eukaryotic cell. These prokaryotic eukaryotic cells contain the cytoplasm, which is said to be semi-fluid substances, which is mainly composed of water. In addition, this cytoplasm contains varieties of elements like cytoskeleton, which forms a fibrous network and provides a fixed shape and support to the cell. The second organelle, it is chloroplast. Chloroplasts are said to be present in photosynthetic cells, which are capable of carrying out photosynthesis. This chloroplast is said to be a membrane bound structure which contains inner as well as outer membrane. The cytoplasmic content of chloroplast is called as stroma, in which the enzymes are present which are responsible to carry out the carbohydrate synthesis. Along with enzymes, it contains a coin-like structures which are arranged into thylakoids, and these thylakoids contains chlorophyll pigment, which is essential to carry out the photosynthesis process. The energy generated through the photosynthesis is utilized in the stroma for the synthesis of carbohydrates. Along with thylakoid membranes, you can also find the DNA of chloroplast, which has the capacity to replicate independently, which does not depend on the genetic material replication. So from, as we can say that mitochondria as well as chloroplast contains a DNA in them. It suggests that these are said to be of prokaryotic origin, which are said to be entered into the eukaryotic cells through endosymbiosis. The next structure of eukaryotic cell is lysosomes. Lysosomes are small membrane bound structures. These structures are generated by Golgi apparatus of animal cells. These lysosomes carry varieties of enzymes which are required for digestion process. So varieties of events like endocytosis, phagocytosis takes place in eukaryotic cells, and these carry either solid or liquid material which has to be treated by the hydrolytic enzymes. So you can say digestive enzymes or hydrolytic enzymes which are required for breakdown of molecules. Like if any cell has taken up the bacterial cell through the phagocytosis, the role of lysosomes is to break down the entire bacterial cell. For the breakdown purpose, it contains hydrolytic enzymes such as lipases, DNases, RNases, then proteases, and so on. So you can find especially this type of lysozymes uh, which are said to be in white blood cells. The next part of this internal structures is vacuoles. Vacuoles are membrane enclosed structures that store materials such as starch, glycogen, or fat, which is required for utilization in the form of energy. These vacuoles engulf food particles, and eventually these are fused with lysosomes for further breakdown. In plants, you can find vacuoles. They are filled with water, and they will help in avoiding the loss of water from the cells. If there is any loss of water from the plant cells, it causes wilting of plant structures. The next structure is cytoskeleton. Eukaryotic cells contains its own skeleton, which is called as cell skeleton or cytoskeleton. This cytoskeleton will form a network of protein fibers which contains three essential proteins. One is microtubules, microfilaments, as well as intermediate filaments. This cytoskeleton provides a support as well as rigidity followed by shape to a cell. Cytoskeleton helps in cell movement as well as cytoplasmic streaming, which is an essential process in eukaryotic cells. Like internal structures, eukaryotic cells also contain external structures which perform essential work for the survival as well as locomotion of a eukaryotic cell. These external structures include flagella, cilia, as well as cell walls. Firstly, the external structure is flagella. This flagella in eukaryotes is more complex compared to the prokaryotes. It contains two central microtubules and 9 pairs of peripheral microtubules. So basically, this flagella is made up of a protein is called as tubulin. The polymerization of tubulin forms a flagella structure. This flagella works like a whip where it will help in locomotion of a cell. As you can see in bacterial cells, the flagella will beat in anti-clockwise or counterclockwise direction so that it can move in forward or it may tumble. So in case of eukaryotic cells, the dynein filament which is located, you can find a dynein filament which is located along with the microtubules help in hydrolysis of ATP. So this hydrolysis of ATP will generate mechanical energy which helps the flagellum to move. So this wave-like motion of flagella will help the eukaryotic cell to move from one location to the another. So you can find this flagella beating or whip-like structure in case of spermatozoa, which is said to be a gamete formed by meiosis. The next external structure, it is cilia. Cilia is said to be shorter and more numerous than flagella, and its chemical composition is same as with the flagella, which is made up of microtubulins, that is tubulin protein. So you can find 10,000 or more cilia are distributed over the surface of a cell. Cilia passes through a stroke and a recovery cycle. So you can find here stroke and a recovery cycle which generates from the base to the tip, which helps in beating of cilia. This beating of cilia help in our respiratory tract to eliminate dust or bacteria. So for this elimination, our body respond in the form of sneezing, or you can say sneezing basically. So this is a basic part of a defense mechanism where bacteria will be eliminated from the respiratory tract due to this mucociliary escalation. The next one is pseudopodia. Pseudopodia is said to be false feet, which is said to be a temporary projections which are prepared within the cytoplasm. So this pseudopodia is basically made up of filaments called as actin filaments. The actin filament polymerization in the cytoplasm will help in the movement of the body of pseudopodia along a solid surface. The next one is cell walls. In case of humans, we don't have any cell wall, but fungi, yeast cells, as well as plant cells contain the cell walls. So cell walls are in algae are made up of cellulose, and some contain other polysaccharides as well. Fungi contain cell walls which are made up of either cellulose or chitin. In some cases it is made up of chitin or both cellulose, or else it is made up of chitin. Chitin is said to be a structural polysaccharide that is common in the exoskeletons of arthropods such as insects and crustacea. And in case of protozoans, you can find a flexible external covering called as pellicles. This structure, regardless of its composition, cell wall gives rigidity and protect them from bursting when water moves into them from the environment. Thank you.